Hello, my name is Chelsea Abbott. I am your friendly neighborhood plant pathologist, and today I will be talking to you through the magic of the internet about wood decay fungi. Let's go ahead and get started. So, first off, what are fungi? Other than being incredible organisms, these are non-photosynthetic decomposers that need to absorb organic matter in order to obtain their energy. So, unlike plants, which can make their own food from the energy of the sun, they're like us, where they need to nom. They need to consume in order to get their glucose and their nitrogen and their amino acids, very similar to us. And fungi are some of the best consumers of all. They go after the hardest material to break down, things like plastics and nuclear waste, or in the case of our wood decay fungi, wood tissue, which is actually very difficult to break down. So fungi are indeed the most common disease-causing organism in plants. This gives them a bit of a bad rap because most of what we talk about within the world of urban forestry, arboriculture, plant pathology, botany, anytime we're dealing with our plant hosts, we're talking about disease-causing fungi. And some of these fungi are primary, meaning that they can attack hosts no matter the stress levels. Or some of them are more secondary opportunistic, meaning these are fungi that come in after the fact. As a consequence, the host has been attacked or is stressed or is wounded, and they are opportunistically taking advantage of that host in order to cause problems. Now, even though most fungi that we talk about are disease-causing organisms, way more fungi out in the landscape are beneficial. And they're beneficial not only to the ecosystem because they decompose materials that are hard to break down, then release those back into the ecosystem for other plants to absorb, but they are also beneficial where they can be mycorrhizal. And so these are fungi that actually have a symbiotic relationship with trees that allow trees to absorb more nutrients and more water because essentially they colonize the root system and expand their surface area. But we're not going to talk about the good guys. We're going to focus on the bad guys, but I just want you to know that these do exist. Fungi with benefits, they're out there. But let's talk a little bit more about fungi in general before we get into the bad guys. So other things I want you to know about fungi is that they exist within a two-body system. So we exist in a one-body system where we reproduce and consume all in the same package. Fungi do this in two separate packages. The first body is the fruiting body. This is the body that produces the spores. Now, spores are the dispersal agent of the fungus. This is how they get around, and this is one of the ways that they get around. And it's really effective because they sort of explode these microscopic objects into the air, sometimes in the number of trillions of these guys. And then because they're very small, microscopic little objects, they're carried by the wind, by the rain, by animals, either short or long distances, depending upon how high up they get into the atmosphere. So this is one of the main ways that they get around, but there are others. Generally, when we're talking about spore-producing structures, we use the term fruiting bodies. Now, fruiting bodies is also mushroom, it's also conk, bracket, crust. There's a couple other names, but if you wanted to be generalized and therefore always right, fruiting body is a good one to use. Now, the problem with a fruiting body is that it only occurs when conditions are favorable, and those conditions are going to be it's got to feed to fuel. So fruiting body takes energy in order to produce. It takes consumption. And so a fruiting body can only really pop up once the fungus has eaten enough in order to fuel the energy in order to take that. So that's one condition that needs to occur. Sometimes fruiting bodies will need to be produced through different spores or different types fungus meeting each other and then sexually recombining to then make this structure, the fruiting structure, the reproductive structure of the fungus. And then lastly, because this is the agent for dispersal and infection, there needs to be conditions conducive to that dispersal and to that infection. So a lot of fruiting bodies and fungi in general tend to like cool, wet conditions, areas where they can really disperse and infect at much higher rates. And so that's why you'll see fruiting bodies of a lot of different diseases, not just our wood decay fungi, coming out at certain times of the year. And it's really because that's the best time for them to, again, either disperse or infect the host. So the important thing to remember is that this might not be there because the conditions have not been met, but that doesn't mean the fungus isn't there. So if we switch over to our second body in our two-body system, remembering that the fruiting body is the one body, not the first body, but one body. This is what grows above ground. This is what often is used for identification. People eat them. People love to play with them. People, whatever you might do with that fruiting body. But remember, there is an entire other second body that a lot of the times is hidden from our naked eye, either because it is literally growing underground or within hosts, but a lot of times is microscopic. And so this is the hyphae, or some people might be more familiar with the term mycelium. So if we're talking about fungi within terms of like plant terms, think about the fruiting body as the flower, the spores as the pollen, and then think about this hyphae mycelium as the root system of the fungus. This is the one that's actually invading, dissolving, and absorbing the glucose. That's going to, again, fuel that host and fuel that fungus in order to do more things. So this is really the one doing the thing, right? Doing the decaying. So always remember that there's a two-body system, one of which might not be there because conditions need to be favorable in order to be produced. Another thing that I want to make sure that we are all on the same page about is when we talk about fungi, we're going to talk about the different types of eating habits. So we're going to have why they eat. Well, they're going after glucose. We've already covered that. What they eat, the saprotrophs versus the parasites. I will discuss that on the next slide. How they eat. This is the white, brown, and soft rots. Again, I will go more into depth of those. And where they eat. This is location, all right? Heart rot versus sap rot, roots, buttress, trunk, branches, all of these are just locations. You will notice in this talk, I am mostly going to be focusing on the buttress as well as the root decaying wood decay fungi, mostly because those are the ones that tend to be the big five of concern, because when a tree fails from a root or a buttress rot, the tree fails versus if it's a branch, or in some cases a trunk, the failure is maybe a little bit less catastrophic. But we tend to focus on the roots and buttress rots when we're talking about the big five fungi. But let's talk quickly about this one, saprotrophs versus parasites. So like everything in this world, fungi exist upon a spectrum. So here is the spectrum of eating of our fungi. On this end, we have our true saprotrophs, also obligate saprotrophs, if you're more familiar with that term. These are organisms that only consume dead matter. That's it. Dead organic matter, that's what they feed on, exclusive. On this other end of the spectrum, we have true parasites, also known again as obligate parasites, and these are organisms that only consume living matter. So these are our two extremes, dead matter, living matter, but you can see there's areas in between. So let's get into it. So this next step up from true saprotrophs are facultative parasites, and I know that this term causes a lot of people like irritation, so I've translated it for you. This is the preferred saprotrophs. So these are organisms that prefer or predominantly eat dead matter, but can consume living matter if, and that's important, if conditions are met for them to do so. And this is most of our wood decay fungi exist in this area. One more step up, we have the facultative saprotrophs. Again, translated, preferred parasites. So these prefer or predominantly consume living matter, but they can consume dead matter if push comes to shove. And this is a lot more of our plant pathogens. So our foliar pathogens and vascular diseases, cankers, excuse me, might exist more in this spectrum. All right, cankers depending on what we're talking about. But let's focus up a little bit more on these two definitions here. So in order to really help you understand, I'm going to give you a human perspective. So if we think about this as far as how humans eat, this might be helpful. If we think about true saprotrophs as fast foodies, fast food, you drive in through the drive-through, you grab your food, you're gone. Pretty easy. True saprotrophs is dead matter. It's matter that can't actively defend itself. It passively can, but not actively. So you're not having to actively combat a immune system from your host that you're trying to feed on. So it's quick, easy. Versus the true parasites, they are actively fighting against a host that is battling back against them. They need to have a lot of mechanisms to evolve around that host defense mechanism. So this is like fine dining. So these ones are very picky eaters, and they're going to be our fine diners. Now, if we talk about these preferred saprotrophs, remember things that predominantly would be a fast foodie, but could be a fine diner if conditions are correct. These conditions would be things like wounding or stress. If we use the example of our eating, if you're a fast foodie, but then you get a Groupon or a discount, and you can then go fine dining. But there's always an asterisk that leads you to be able to be a fine diner. You can't just be a fine diner out of nowhere. There has to be a condition met. And then if we go one more up, we have our preferred parasites. So again, these are going to be our preferred fine diners. But imagine we're on a long road trip, just exhausted. They'll absolutely grab some fast food. You know, they're not going to turn their nose up to that. But it's again, typically most in this category will be on a living host, kill that living host, and then feed on the dead remains. So in general, this is the spectrum of what fungi eat. So I just want us to understand more specifically that most wood decay fungi are actually not parasites, true parasites. They are more this liminal space in between saprotrophs and parasites. And to put it further in perspective, allow me to give you some examples of some diseases that you may be aware of that fit into these categories. So some true parasites or obligate parasites that you may be aware of: the rusts, powdery mildew, and viruses. These are diseases that only feed on living hosts. Versus some preferred parasites that you might know: weeping conk, chicken of the woods, Armillaria root rot. These are ones that they absolutely can feed on dead matter, but they can feed on living hosts, particularly hosts that have been stressed out or wounded. Stress and wounding helps all of our wood decay fungi and most diseases in general. And then some preferred saprotrophs that you might know. Again, these ones are going to be saprotrophic leaning, but can be parasitic, particularly when we have that condition met of stress, wounding, or other factors. And these are going to be artist conk, brittle cinder fungus, varnish conk, hen of the woods, oyster mushroom, split gill fungus, dead man's fingers, bisconeoxia, aka formerly ipoxylin canker. Now, the lines between these two can get a little bit blurry, and a lot of times we don't differentiate between a preferred parasite and a preferred saprotroph. Most of what I want you to know based off of this is the ones that are highlighted in green are the ones that we typically talk about when we talk about wood decay fungi. And that's because they have been known, a few of them, weeping conk and Armillaria, as well as brittle cinder fungus, have been known to breach the barrier and reaction zones of living hosts, which means that they are able to be more parasitic and they just don't stay in the dead wood, aka the saprotrophic area within the host. But there are some true saprotrophs that you might be familiar with, things like dryad saddle or pheasant's back, carbon balls, dog vomit, which is technically a myxomycete or a slime mold, inky caps, which are little tiny mushrooms that sort of dissolve into black goo. These are saprotrophs, and the jack-o'-lantern fungus, something that looks very similar to Armillaria root rot, but clearly is quite a different type of fungus. So again, these are ones that are going to exclusively stay in the dead wood and not be able to breach past that barrier or reaction zone of a living host. And so they just would feed, for example, just in the heartwood, whereas some of these other ones can breach into the living tissue, aka the sapwood. And so again, mostly what I want you to understand about this is that they exist on a spectrum. Some of them can be parasitic, some of them can be more saprotrophic. Not all fungi are created equal, and some of them are even more pathogenic, meaning they're able to cause more disease than others. And partly it is because of this reason. But let's talk about where these fungi would feed. So generally, a lot of the wood decay fungi that we talk about are heart rotters or heartwood decayers. Now, I know heartwood is a bit of a charged term. Some people don't like it because not all trees produce heartwood. So we can just go with heart rot, and hopefully, basically what I'm talking about is the central area of our trees in the programmed cell death area. So this, even though these are programmed or cells that have been programmed to die and therefore are dead, this does not mean that they're not resistant to decay. In fact, there's a lot of passive defense mechanisms that have essentially been left inside these cells to be resistant to decay. Think of them like the tree putting up landmines, so that these heart rotters, these fungi that would predominantly decay in the central stem, won't be met with no resistance. And these are going to be things just by the environmental conditions, very low oxygen conditions, and a high amount of defensive toxins and inhibitory substances that have been literally just pre-programmed into the cells themselves. True saprotrophs can only attack the heartwood, the dead cells within the tree, unless of course the sapwood becomes damaged, and then they can move into that area if the sapwood has been dead. But there are the more parasitic leaning fungi that can decay in the heart rot, but again, actively breach some of those barrier and reaction zones and become pathogenic moving into the sapwood. And again, those are going to be more of our pathogenic leaning fungi like weeping conk and Armillaria and Kretzschmaria deusta. Now, stability overall related to a heart rot is less concerning because especially if it stays central. So a centrally hollowed out structure that doesn't breach a certain diameter is actually quite stable. The issue is when that internal hollow exceeds a certain diameter or circumference in relation to the diameter or circumference of the tree, or if it becomes offset. Once it becomes offset, the area it covers is less. But if it stays central and it does not exceed a certain radius or diameter or circumference, then generally a heartwood decay is less concerning from a stability standpoint. And depending upon the pathogen, heartwood or heart rot decay is typically a slower decay strategy. One of the other big things that people will say about heart rots in general is that if you have a singly or large singly growing fungi growing up a tree, that's more indicative of a heart rot. Versus a sapwood decay or sap-rot, it's a much more tiny little guys all over the tree. And I'll show you a picture of that in a second. So here you go, a lot of tiny little fungi all over, numerous tiny fungi, sap-rot, singly large fungi, heart rot. Not exclusive, but it's a guideline. So with sap-rot, this is the living part. This is the external part of the, or right next to the bark part of the tree. And so these are the living cells that are actively moving around water and nutrients within the host. And so anything that tries to decay the sap-rot must deal with the active defense mechanisms of the host. This is where the host and the fungi are having a battleground. And things can be like with their compartmentalization, but also things like tyloses, gum deposits, certain phenolic fungistatic compounds that are literally, again, there to defeat the fungi. And then just chemically thickened cell walls that will help resist decay. So this is more of an active battleground, whereas the heart rots are more of a passive battleground. So if the sapwood is damaged or dead, then the sapwood, or saprotrophs, can invade, but more parasitic fungi can invade without the damage stress. Again, I've already noted which ones are a little bit more aggressive. Generally, sapwood pathogens have a more rapid invasion strategy. They're going to colonize the wood extensively and then cause decay, particularly when the wood has less water content. There have been some studies that show that the turgor pressure, the minute it drops from, say, drought stress, these wood decay fungi are able to penetrate into the cell. But as long as that turgor pressure stays up, that water potential is enough to keep them out. So having a lot of moisture in the sapwood is a very good defense mechanism along with all of these other things as well. But of course, when we're dealing with things like drought, that's when we can start to see a lot of these pathogens invade and colonize a lot of our hosts. Typically, there's more strength loss occurring proportionally with a sapwood or sap-rot than there is a heartwood rot or heartwood decay, heart rot. And that's because, again, you're getting very offset. So that internal decay is on the edge, and you see a lot more strength loss because of that. There's also generally a good amount of decline associated with a sap-rot. just because, again, that's the living tissue. That's what's actively moving around water and nutrients. And so once you start to damage that, you do see some symptoms of decline. Now, that's sort of the where, the location. Let's talk about how fungi eat. And this is going to be our types of rots. So we hopefully at this point know them. I'm going to get into depth, but I'll just preliminarily give you an overview. We have the white rots, which is the most common decay fungi, about 80%, depending upon where you're at. If you're in an area that is way more conifers than it is deciduous trees, this number might not hold up. But particularly if you are in an area of more deciduous trees, you're going to see a lot more white rot. Again, usually 80% of the time, it's going to be a white rot. Versus a brown rot, now these ones are less common. It's more common, again, if you are in an area with a lot of conifers. But generally, overall, we don't see a lot of brown rots, particularly on the deciduous trees. There's a couple, and I will note one of them. But for the most part, it's less frequent that you will come across a brown rot. And then a soft rot, which is visually, if you're staring at it, more common or more like a white rot from its looks, but chemically it acts like a brown rot in the way that it degrades the cells. And so we use these terms to talk about wood decay fungi because it's important to understand what they're doing to the tree, because same thing as sap rot versus heart rot has different levels of strength loss associated with it, as do these. So before we dive in further, I want to just quickly talk about what the structure of a wood cell is. So a well-formed wood cell is going to have both flexibility and rigidity. And that's because you need tensile, tension strength, and compression or compressive strength. You need both. Both are incredibly important. So I like to think of cellulose as the ropes of our cells. It's stringy, but you can really pull it, right, and get that tensile strength. Whereas lignin is going to be more like our bricks or our concrete. That's going to be something that's very rigid and good for compressive strength. Again, needing both. Typically when we're talking about cellulose, we're talking about it's mostly within the outer and inner layers of the cell wall, and then lignin is kind of wrapped up around in between it. And this will come into play when we talk about how they're degraded. But always remember, cellulose is ropes, good for tensile strength. Lignin is bricks, good for compressive strength. So let's talk about white rot a little bit more in depth. So white rot, we have, oh my goodness, white bleached appearance to the wood. I say this all the time. Scientists, we're not very creative people. We tend to name things what it looks like. And this will become very apparent when we do our wood decay identification, because you'll start to notice that a lot of the common names of wood decay fungi kind of are telling you key identifiable characteristics of that fungi. So white rot leaves behind a white or bleached appearance to the wood. There is technically two types of white rot. The first type is something called simultaneous white rot. This is where both the lignin, the cellulose, and the hemicellulose, which people don't talk too much about, break down at the same rate. So you end up getting a very soft and brittle tree at the same time. The second type is selective delignification. This is where lignin is first degraded and then cellulose comes at a later stages. This is what most of our white rot, or most people who have dealt with white rot will think of. It's a little bit less rare as the simultaneous white rot. However, it's good to note that white rots can kind of flip back and forth between these two, particularly in advanced stages. Okay. So one of the things with white rot, again, or most rots, is it's not necessarily a cavity sometimes that's formed. It's actually just the quality of the wood is affected. So in this instance, you can see this tree was tested. It was found to have very low quality, and it came up like it was having cavities, no resistance, that kind of thing. But when a tree was cut down, you're like, oh, but there's wood there. But go ahead and just stab it. And you'll see that it's punky, it's stringy, it again has that bleached appearance. So sometimes the wood might not be fully gone, but the quality is gone. So it's always important when you're doing any type of assessment with tools, you are testing the quality of the wood, not necessarily the absence of it, okay? All right, so let's talk more specifically. White rot, generally we have significant strength losses in the more advanced stages of decay. This is different from brown rot, which we see more strength loss in early stages of decay, and that's one of the separations between the two, as well as what they do to the tree themselves. So wood with simultaneous rot has very little strength overall because, again, it's taken away the ropes and the bricks. Everything's gone. Whereas wood with selective delignification has very little compressive strength and okay tensile strength to advanced stages. And that's because a white rot specifically, as the name would suggest, is going after the lignin first, cellulose later, which means we lose a lot of the bricks and we're left behind with a lot of the ropes, hence the super stringy looking rot as it decays. And so you don't have a lot of the compressive strength, but you still have some good tensile strength, unless we're talking about simultaneous rot, which takes away all of it. All right, so generally, our white rotters is our most common. I would say, again, most of it is going to be a selective delignification unless otherwise noted, okay? Now, if we flip over to brown rot, so brown rot is brittle and altered lignin. So mostly it's going to go after the cellulose and the hemicellulose, and they'll leave the lignin again for more advanced stages. Now, typically there's significant amount of strength loss in the initial stages or incipient stages of decay. So that's like very, very, very beginning stages. And so while we have with our white rot more strength loss in advanced stages, brown rot, you see more strength loss in the earlier stages. And so that's a bit of a difference. Again, just visually, you can tell the difference between a white rot and a brown rot because the wood is brown. It also tends to be very crumbly. So remember, we're taking away the ropes, we're leaving behind the bricks. So a lot of the times, the wood just kind of looks like a bunch of crumbly bricks. All right. Now, when we have our strength loss in general, we have that cellulose taken away, so we lose a lot of our tensile strength, but we have okay compressive strength. Again, in advanced stages, it kind of loses both. And a great example of this one would be the chicken of the woods. And then our last type of decay. For a very long time, soft rot was sort of thrown in with white rot, but of course, now we know it is a different type of rot altogether. So soft rots, chemically similar to brown rots in that they attack the cellulose. So they're attacking the ropes. They're leaving behind the bricks. Eventually they get the bricks as well. Eventually they'll more specifically move through the cells rather than degrading them. They create these grooves and tunnels within the cell wall itself. So a lot more cavities form. And I'm not talking large cavities, I'm talking like microscopic cavities form within the wood with a soft rot versus a brown or a white rot, which would degrade it a little bit more like an erosion rather than punching through the cells like a soft rot does. This is a huge loss of tensile strength because you are losing that cellulose and leaving behind the lignin. So again, even though visually it looks more like a white rot, it acts more like a brown rot. And so the best example of that one is Kretzschmaria deusta. There's a couple other soft rots, but this one's the big one. Okay, so that is sort of our dense information about wood decay. But let's get into our actual mushies. And so in order to prepare for such an event, to look the part, I shall don my mushy hat. And let's talk mushies. Okay, so now we're going to do actual identification. Ready? 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 I'm mostly going to be focusing on buttress and root rotters, and I won't necessarily be focusing a lot on trunk, branch, sap rotters, or any saprotrophs. And again, that's because most of the times when we consider the wood decay fungi that are the most concerning, we're concerned about the root system and as well as the buttress system. But I don't want to discount concern. If you find fruiting bodies growing on a host, it's always a red flag and needs further investigation. So if you see a branch covered in fruiting bodies, it's a pretty good chance that branch is decayed and you should not be tying into that. So just FYI, it's not to say that the branch and trunk fungi are not dangerous. It's just that these ones are more concerning generally for tree failure. So which ones am I going to be talking about? The parasitic leaning fungi, of course. I have a whole other talk on saprotrophic fungi. That again, these usually can be seen in pairs, I would say. Meaning I'd love to talk about the saprotrophic fungi, particularly the ones that look like the more parasitic leaning fungi. But for this talk, I'm only going to be focusing on the parasitic leaning fungi in that spectrum of eating that we talked about. Now I have ordered these specifically. I've ordered these as far as what I consider to be, let's say, least to most concerning. Now I want to be clear, that does not mean that Varnish conk is not concerning. It's just a relative situation. And there's a couple reasons I've used to organize them thusly. How easy are they to identify? The ones that are harder to identify go lower on this list, simply because people will miss them. In some cases, fully not identify them at all. In other cases, maybe misidentify them for something else. What type of rot do these cause? A lot of these are white rotters, except for chicken of the woods and brittle cinder fungus. And so a lot of these are going to cause that stringy type of spongy wood versus this more brittle wood. And then where do they decay? Generally, I would consider more root rotters to be more concerning, because not only is it what's happening to the tree, but it makes it much more difficult to understand how much of the tree has actually been decayed. Unless you do like a root crown excavation, you really can't know with some of these fungi that are lower on this list. All right, so just keep that in mind. Again, I'm not saying that some are not concerning. It's just a relative thing. So, before we get into the identification, let's make sure we're on the same page about some of the terms that I may or may not use. So, is it flat, or if you've ever heard recipienate? Is it a mushroom? Now these are going to be fleshy annual. Think of things like in the grocery store. Is it a woody bracket or conk? Generally, brackets or conks are going to be more shelf-like fungi. So you can really have just like growing on a tree. I am the tree. And then you could put something on top of it. It's a shelf fungi. All right, so that's sort of a woody bracket. If you had a mushy and you were trying to figure out what its spore side is, you would flip it over and you would look at it on the underside, generally the side that's facing the ground. And so this is the spore side. And now this side can have gills, pores, spines, teeth, or none. There is one that we covered that doesn't have any of these, and those are called crust fungi. But yes, we would be looking on the underside, the spore side, and we would be looking again for gills, teeth, spines, pores, yeah, okay? And then the cap. So the cap is typically the part that faces the sky, okay? What are the notable textures, the colors, the patterns? Does it have any guttation or weeping points? Guttation is just like a funny word for sweating. All right, so any type of notable texture, color, etc. Again, color is going to be important not only on the cap, but also on the underside, on the spore side, on the stalk. And then another term that I want you to know is the context. So if I say cut this in half and then opened it up like an apple and you were looking at the internal part of it, that's what we call the context. And there's some wood decay fungi that you need to look at the context in order to identify. So just so that we're familiar with what that term is. Does it bruise after being handled or cut? And now be careful, because sometimes it takes like 15 to 30 minutes for it to bruise or oxidize in order to change colors. So if you do go this route, if you're trying to figure that out, some of them will bruise instantaneously like that. Some of them take some time. So you might have to wait depending upon which fungus we're talking about. And if it does, what color does it turn? Okay. What time of year is it? Wood decay fungi are very punctual. They like to come up during certain times of the year, and so I'll try to remember to note when typically you would see this wood decay fungi. Obviously, it would be different depending on where you are, but I'll give you sort of a general timeline. There is a cadence to wood decay fungi, so knowing your time of year. For example, Armillaria will not fruit in December or January. So if you see something that looks like Armillaria and it's January, it's not, okay? And then just always remember, it could not be present Remember, we talked about how wood decay fungi, the fruiting body is for producing spores and reproduction, but the eating body, the vegetative body, is that individual, that root system that is inside the host or in the soil. And so this does not necessarily need to be there in order to have the fungus doing any decaying. So this is just helpful for us, okay? Not necessary. Okay, let's talk about our wood decay fungi. Let's start off with Varnish conk, aka Ganoderma sessile. All right, so this one's going to be mostly on the hardwoods, again, depending on your location. I find it mostly on the oaks and the honey locusts, but it can be on plenty of trees. It is a white rotter selective delignifier, so that means it creates that super stringy rot where it's mostly leaving behind the cellulose and getting rid of the lignin. So you get that really like stringy loss of compressive strength within the tree. It is a root and buttress rotter, so there has been some more evidence that it works in the roots, and within the buttress system, the decay, at least in my experience, is fairly localized to where the actual fungus is itself. But again, it's hard to know sometimes with root rotters how much decay is actually occurring unless you have either probe into the root system or you do a root crown excavation. As far as like its identifiable characteristics, Varnish conk is pretty good for you to remember. Conk meaning it is going to be a shelf-like woody sort of a mushroom or fungi, so that when you touch it, it's not going to be super fleshy. It's going to have some toughness to it. And then varnish, it has a shiny top. It looks like it's been varnished. If it's in its textbook version of itself, it will be kidney bean color and kidney bean shape. So nice red mahogany, shiny top with this really great white creamy margin. Sometimes it will stack up upon itself and it can dye itself some colors. Its spores are like mahogany colored, so it can sometimes lose this white margin. But one of the issues with this fungus is that it's an annual fungus. So an annual fungus is something that produces this fruiting body all in 1 year. All right, so you can find it at different stages within its yearly life cycle. So when you find it when it's young, it looks more of like a spore mat. So you kind of get a lot more of that spore side, that underside. And now it is pores on the underside, so that's helpful. But a lot of times you're just looking at it and it could look like a lot of different other fungi, so it gets a little bit hard at this stage. But I will say, even in this young stage, when it's mostly spore, you should still be able to find areas that has that more mahogany color and in some cases the shine. And so that's a good way to try to find it. Or try to identify it at these stages. It should always have like a little bit. You might have to go looking for it and you might have to turn it over and look at the underside, but you should be able to find at least a little bit of that part that has this more plasticky feeling, red mahogany color with a shiny top. And because it is an annual fungus, another really important thing to do is to look at the spore layer. And so when we crack it in half, break it in half, you will notice that there is only one spore layer. So right here, this is the cross section of the pores. So they look a little bit like spines a bit, but they're not. Spines would be detached at the bottom, at the end here. So there's only one layer of that, and that's very important because its other sister, the Ganoderma applanatum, is perennial. And that's one we'll cover next. So artist conk, aka Ganoderma applanatum. So again, hardwoods. Find it on a lot of oaks, but wide host range. I've found it on beech, I've found it on maples, I've found it on honey locusts. This guy gets on a lot. All right, now this is a simultaneous white rotter. So now we're getting into the complete loss of strength, compressive and tensile. And it does tend to go in mostly within the buttress system and a little bit into the roots. It can spread through the root grafts. So if you've got a bunch of oaks that are root grafted, there is a way for it to get from oak to oak to oak to oak via those root grafts as well. But generally, a lot of the decay is mostly within the buttress and trunk system, typically associated again with where that wood decay fungus is actually fruiting. All right, but again, and that's not exclusive. I don't want you to live and die by that. Now, in order to identify this guy, it's called artist conk for a reason, and that's because it bruises incredibly easily. You can actually see my fingerprints from pulling this off, it bruised. And that bruising will stay forever. So be careful what you write on these guys. But when you pull it off, and you can just lightly drag your finger, or again, even just pressure from your fingertips, and it will oxidize dark brown, and then that will stay forever, hence why it's called the artist conk. But you could bruise anything if you try hard enough, and if you're in the dead winter, or if you wait over 48 hours after you pick this off of a tree, the bruising won't work anymore. So here's some other identifiable characteristics. It is more hoof-shaped, so it's much more rounder in its shape. It is still a shelf, so it is still put a little thing on top of it. So it's still going to be a conk, very, very woody to the touch. It's not something that you're going to be like, I could cook this up. No, you will not think that, and you shouldn't. All right. It will also have these really beautiful ridges, a more tannish top, not so mahogany colored, but again, it can stack up on itself, and its spores also are mahogany brown, so color might be a little bit tricky depending on when you get it. And it has this white margin, and again, white underside, which are pores. So pores again on the spore side. Now, generally, my best way to identify that, you can use some of those colors and the difference in the ridges. Again, you can see here between a Ganoderma sessile, the Varnish conk, and then the Ganoderma applanatum, there's a little bit of a color difference. But I believe the best way to identify this fungus is breaking it in half and looking at the spore layers. So remember how I said Ganoderma sessile was an annual? Ganoderma applanatum is a perennial. So it builds upon this fruiting body every single year. So you can count the spore layers as such and, see that it is, let's say, 1, 2, 3, 4. This conk is 4 years old. This conk is 1, 2, 3, 4, 5, 6, 7 years old. Now, that does not mean that the infection is 4 years old. It means that it is at least 4 years old, or at least 7 years old, right? Because remember, this was only produced when conditions were correct, okay? So that's the best way to identify Ganoderma applanatum. Now, this one is more aggressive as far as the rate of decay, as well as the type of decay. It's not a selective delignifier, it is a simultaneous rot. So generally you see a lot of decay associated with these guys. And if you were to sound it with a hammer, you could typically hear quite a bit of a hollow, okay? All right. Now let's flip over to chicken of the woods. So chicken of the woods, Laetiporus sulphureus. Now keep in mind, depending upon where you are in the world, you might come across different species of this. So there is a cincinnatus version, which is slightly like if you turn the saturation on my computer down just a little bit, you would get more of what the coloration would be with a Laetiporus cincinnatus. And there it would be more like pinky, salmon, and then cream on the outside. And then there's conifercola. I think I'm getting that species name right. That's going to be more of the Laetiporus that gets on conifers, and that's more common out west. Okay? But this one specifically, this one's mostly found on oaks, cannot be on many hosts. Generally, when we find it, or at least when I find it on the trunk and the branches, typically on or near cavities and cracks. So a lot of times I'll find it either with a crack or it'll be inside a hollow from an old wound or something like that. The really cool thing, the fungi fact about this guy, is that it can deposit resting spores within its host. And so there's actually some studies that have been done to show that some wood decay fungi can live as endophytes, and they're inside the host without causing any damage at all, but they're waiting. They wait until conditions are correct for them to infect, and then they attack. And so there's this paper, I think it's like, do trees hold the seeds of their destruction? And it's about how certain wood decay fungi can just kind of hang out in a host, essentially just waiting for the host to get stressed or weakened, or for the wood to dry out a little bit, or for other conditions to be correct for infection, and then it'll flip into a parasite. Now, I noted that there are other types of Laetiporus, but in general, this is how you would identify it. For sulphureus in general, the color is going to be probably your biggest indication that you're dealing with chicken of the woods. That is, you have bright orange cap with a really sulfur yellow margin, and underside that is a poor spore side. I'll show you some photos of that. Now, if we were talking cincinnatus version, again, that orange would just be a little bit toned down, and so would the yellow, but these other identifiable characteristics still hold. Generally, chicken of the woods typically will be more of a shelf-forming fungus. The Cincinnatus version, I do tend to find more of like a bouquet at the base of the trees. But if you look at the actual like individual fungus itself, it will be very fleshy because this is a choice edible. This is something that a lot of people will consume. And so it's very, very fleshy, kind of like almost the consistency of like firm tofu. And so it's not woody, it's a very fleshy annual fungus. The spore side or underside is going to have very tiny pores, very, very tiny. It will look like velvet. And that's key because there's a lookalike that has much larger pores that is very different from this fungus. It does cause a brown rot, so you're going to lose a lot of your tensile strength. And excuse me, you're going to lose a lot of your tensile strength with your decay pathogen here. A lot of the times I see trees fail from this fungus before they die. So it's kind of like one of those things where they'll sort of break, all right, because they can become very brittle. All right. And then one of the other identifiable features of this guy, again, other than just being an annual, some people will say that it has a very acrid smell to it. Very like sulphureus, it's a very almost like rotten egg type of smell. Odor's not a great one to go off of, but you know, I throw it out there just in case you come across it. Okay? All right. So that was brown rot. Now we're back to the white rots again. So we're back to having a big loss of compressive strength and tensile strength again until advanced stages. Okay, so this one is one of my favorite wood decay fungi. This is the weeping conk, aka Pseudoinonotus dryadeus. So this one, I love it mostly because of the way the fruiting body actually looks. But before I get ahead of myself, let's talk about where it is. It is a roots predominant white rotter. And so that's unfortunate because, again, generally with this one, it works its way from the bottom to the top of the root. So by the time it gets into the buttress system, it's typically assumed that the root system has been decayed. Now, this is not necessarily true of all root rotters, just this one has been observed to have that type of decay. Now, as it moves through the tree, it does produce the fruiting bodies seemingly the later stages of the decay. So seeing the fruiting bodies is also an indication that there's probably a large amount of decay associated as well. So again, that's not a blanket statement of all wood decay fungi, but this one in specifics. Now, because it is a root rotter, it can kind of change its, I would say, look depending upon where it's on the tree. So if it's out in the roots, it has more of this amorphous blob-like look to it, very creamy surface, and then this really conspicuous amber droplets weeping. All right, it's called the weeping conk for a reason. So this is guttation. One of the theories of why it guttates or causes this sweating or this moisture is because it literally broke a sweat from creating a fruiting body so fast. It's like a metabolic energy thing. Some other people think it's to attract insects to help with spore dispersal. I like the sweating theory just because I just think it's funny. It is an annual fungus, so it can degrade over time, and I generally see this one popping about June-ish time of the year. That's when I'll start to get pictures, sometimes a little earlier, sometimes a little later, depending upon the temperature, but it does tend to come out in midsummer. And then by the end of the summer, it starts to degrade. So typically, again, if you see it out in the root system, exclusively fruiting off of the roots, it'll look more like this blob. But if you see it on the buttress system, because it can be a buttress rot as well eventually, it'll be more of this like conk-like, where it's more shelf. Again, it's going to be a little bit more squishy, but pretty tough to the touch. It's not something that you're going to think, this should be edible. It's not. Don't eat it. It will sometimes have more of a tannish color to the actual or the color of the fungus itself, but that amber droplets are pretty indicative. I have seen them be a little bit clear, but generally I see it as amber. Most of what it gets on is oaks, but I have found it on maples as well. So keep that in mind. I would say generally more often than not, it is a oak fungus, but it's not exclusive. This is what it looks like as it degrades. So again, it can get more difficult later in the season, later in the summer and into fall. It starts to look a little bit more like a Ganoderma sessile. If you are confused which one it is, always assume it's the more dangerous one, which in my experience this one has more decay associated with it, so I would assume it was this one. Now, throwing a wrench into your whole plan here. Oh, the weeping conk. Look for weeping pores. Sometimes it's too dry. So you can see on this sample here, you can see the craters where the weeping ports and the guttation was, but it's too dry outside, so it actually does not have the moisture, and it essentially evaporated. In those instances, I do recommend you to rip this guy off and break it in half and look at the context. Not only will the context weep, but you'll see these sort of like layered rings, and it will be brown. And this is very important because there is a look-alike fungus that is white marbled on the inside. The other notifiable feature of this is when, again, you look at the context, you should see a pretty good contrast between that sort of the cap coloration, so this yellow line, and then again that brown internal, and then just one spore layer. Okay, it's an annual. Okay. All right, moving right along to Armillaria root rot. So Armillaria root rot, huge host range. Generally, it is a white rot, simultaneous, so that's loss of compressive and tensile strength, but eventually it can become a soft rot. It can spread through the root grafts, so this one we know for sure will spread from tree to tree to tree via the root grafts. But one of the other things that it creates within the wood as it decays are these things called pseudosclerotial plates. Now these are these zone lines, they look black, and essentially what this is the fungus marking off its territory within the host of where it's going to decay. So it's not only to keep other wood decay fungi out, but it's also to keep the conditions of that wood appropriate for its decay. So if you see these zone lines, it might be Armillaria root rot. There's another one that causes that. Well, there's a few, but two in this presentation that cause that. Armillaria root rot mushrooms are incredibly difficult to identify, but I'm going to give you some key features of what you would be looking for if it was Armillaria. Armillaria is always in a bouquet form. It's always in a cluster. It is not a singly growing mushroom. So you should be able to find the common root that all of the fungi are growing out of. If you don't find that, then it's probably not Armillaria. Again, it is not a singly growing mushroom. Generally, it's going to be more tannish, sometimes a little bit yellowy, never orange really, on the cap, and then the stalk as well as the gill layer or the spore side are going to be a more creamy tan color versus the top. Again, bouquet form, it is a root rot, so like Pseudoinonotus dryadeus, it can be sometimes found out in the root system, sometimes up against the buttress system. Another identifiable characteristic though, unless you're talking about ringless Armillaria, which is more rare, you will find these rings around the stalk. And this is where the fungus used to connect to the stalk, and then it rips off and it leaves behind this almost like veil, which leaves behind a ring. And so that's an identifiable feature as well. So look for clusters. Typically I will find this fruiting in August, if at all, and then look for those rings. And that should hopefully help you identify it. It also has a white spore print, but generally I don't see this fungus fruiting. More often than not, I see its other signs. So Armillaria root rot is also called the shoestring root rot, and that's because it creates these things called rhizomorphs. So rhizomorphs are essentially specialized melanized hyphae conglomerated together. And so remember, hyphae are the vegetative root system of the fungus. So this is the one that's actually doing the eating. So rarely will you see the mushies pop, but the rhizomorphs should be there. You might have to go looking for them, but they should be there. They're either in the soil, growing on top of the host. They'll look more like plant roots in that instance. Crack them in half, and the coloration will be consistent throughout. If they're growing underneath the bark, they're more flattened. And they're growing under the bark, you're like, that's not a plant root. Sometimes you'll find the mycelial fans or mats. You'd have to go digging for that, and sometimes it depends where you get it in its infection cycle, so it's less reliable. Here's some more images of rhizomorphs, since I think these are the most likely form of Armillaria root rot you would come across. Again, flattened, shiny, melanized hyphae growing underneath the bark system or sometimes on top of the bark, depending upon where the host is. If you're really trying to figure out if it's a plant root or rhizomorph, sometimes you can gather the rhizomorph, put it in a plastic baggie with a little moist towelette, and it might actually produce some hyphae for you. And that's a way to tell that it is not a plant. I've only been able to do this a couple times, but it might be a helpful tool, particularly if you're trying to figure out if this is plant roots. This one I hope you wouldn't think, but you might be like, I want to check this out. But yeah, typically when we see rhizomorphs, the infection is pretty far, and we will see canopy decline. Yeah. Okay, now, if you've forgotten everything that I have said in this presentation, please remember this one. This one's the big bad one. This one's the one that I want you to walk away with, if nothing else. So this is Kretzschmaria deusta, aka the brittle cinder fungus. Now this one has a crazy wide host range. I found it on pretty much everything. Depending on where you are, it might look like certain hosts better. But again, maples, beech, hickories, catalpas, oaks. I feel like I'm missing a bunch, but my point is many hosts can get this. Now, it is a soft rotter, which means it creates that brittle tree because, very similar to a brown rotter, it's going after the cellulose and leaving behind the lignin, but then eventually it does sometimes turn into a white rot, so then you get a just full strength loss throughout the board. All right, it is a root and buttress system host or targeting, so typically if a tree fails, it fails right at the base, and you can kind of see that right here. And it does create those pseudosclerotial plates, very similar to Armillaria root rot. The tricky thing with soft rots as well as brown rots that I forgot to mention when I was talking about the chicken of the woods is if you were to sound this tree with a hammer, it doesn't necessarily tell you how much decay is actually has occurred. Same thing with a resistance drill and some types of sonic tomograms. The data from testing soft rot and sometimes brown rot is not actually true to what the reality of the decay is. And so you have to use a certain type of tomogram, or in a lot of cases, it's identifying the fungus only. And not only is it high on this list because of the type of decay it causes and therefore brittle tree and location, it's mostly because I find this one oftentimes gets completely missed or misidentified. And that's because it is a crust fungus. And I'm saying crust fungus not only in the way that it actually is formed, it looks like a crust, but it also doesn't produce any spores, spines, pores, spines, gills, teeth. It is just a flat structure. And so it is a 2D-like growing structure that grows up the side of the tree. So if there's ground cover or snow, it can often go hidden. But then also it's hard to identify in its stage that it mostly exists in. So in like May, June, it'll be a little bit easier because it'll be more velvety, it'll be grayish with a whitish margin, and sometimes they've even found it weeping amber liquid or guttation. Okay, this is very easy. Again, if you were to touch this, it's velvety soft, but about at the end of June, it'll start to turn black. And then it'll start to form into its, what I find most common looking structure, which is this warty black charcoal-like tissue that is just growing on top of the bark. And so a lot of times people might mistake this for wetwood, literally discolored bark, or again, they'll miss it because it's covered by something or the color is not as distinctly different, and it's not a 3D shape, it's 2D, flat to the surface. A couple things I'll have you do in order to identify this is crack it off. You'll notice that there is bark beneath it. If you try to break off bark that is just wetwood, you'll be breaking off bark, right? This one you are breaking a fungus off and the bark is underneath. The other thing to do is to look at the surface of this warty tissue, and you should be able to find these black pimple-like structures. This is actually where the spores are coming from on this stroma. And so these black pimple-like structures are pretty indicative of this fungus, as well as, again, the triangle-like location or the triangle-like growth pattern on the host, typically touching the soil line, except for one host, beech. And it's warty and almost looks like lava rocks, and it'll be covered by these little black fruiting bodies. And so, as I mentioned before, beech is the one host that kind of bucks that. Typically, I'll find it growing next to the soil line in a triangle. Beech, I usually find it in the crevices, in some of these like furrows, and it's like hidden in this crack. So with the beech, just make sure you're kind of checking in the furrows, not necessarily looking next to the ground. And one of the cool maybe cool things is that I've also found it inside trees. So here it is inside the internal cavity of this beech. So be aware that apparently that can happen. Again, it just needs to have an air current in order to spore disperse. So technically, I suppose it can fruit in cavities as well. All right, but always look for this flat-like structure. If you can see it when it's young, it has that, again, gray velvety look with the white margin. Otherwise, it's going to exist more in this warty, blackened lava rock with tiny little black fruiting bodies on it. And then the last thing that I'll have you be aware of is the fact that these fungal pathogens can indeed partner with each other and then have a sort of succession. So that means I have seen in cases when a lot of these fungi will actually come after one another. Because again, remember, weakening the host or creating wounds is a precursor to some wood decay fungi infection. And so, for example, I've seen like Ganoderma and dead man's fingers working together. On beech, I see a lot of Ganoderma applanatum as well as Kretzschmaria deusta working together. And in some other cases, I've seen a saprotroph, full saprotroph, like a slime mold, working on the dead spot created by, again, another Ganoderma or wood decay fungi. So there is sometimes multiple species, a lot of times multiple species working on one host. So if you find one, maybe do a double check to make sure you don't find any others. But that's it. I hope that that was very informative for you. If you need any help identifying wood decay fungi, or if you want to ask any questions about this presentation, there's a couple places you can find me. Here's my email, as well as I am online and social medias. More than happy to discuss any wood decay fungi or fungi in general if you need so. But I hope you had fun and stay curious.